This video is about CRISPR. CRISPR is a really incredible tool that scientists use now to do gene editing that makes all that restriction enzyme stuff that we talked about in our last unit seem just like a very, very crude tool. So CRISPR is very much like the restriction enzymes we talked about in our last video. CRISPR is a system that bacteria have in place to protect themselves from viruses, but we use it now in order to do biotechnology. And it's just a much, much, much more powerful tool. So what does CRISPR stand for? CRISPR stands for clustered, regularly interspaced, short palindromic repeats. So this is bacterial defense to viruses. So here's a bacterial cell, and here's a virus that's injecting its DNA or RNA. A lot of times when a virus injects its DNA into a bacterial cell, the bacterial cell dies. But sometimes the bacterial cell is able to recognize the virus, and maybe it has a restriction enzyme or something, but it's able to defend itself and live. And when it does, it will take this viral genetic material and it'll stick it here in its very own chromosome. So this is kind of a crazy thing. It's taking its enemy, its virus DNA, and it's putting it in its own chromosome. So it's not putting it in the chromosome in order to use that DNA. It's putting it in the chromosome to say, hey, this is a bad guy. If we see this again, we should do something about that. We should fight it off. It's almost like if you go into a police station and you see on the walls, or sometimes in the town hall, they have this like the a wall of like criminals they're trying to find, right? Here's the mugshot of the person we're trying to find. It's a little bit like that. So here's a virus, DNA from a virus that's infected this cell before, and here is the some DNA from a virus that's infected this cell before, and viral DNA again. But they're separated out by these short little repeats, and so these are all clustered together, all the repeats plus the viral DNA. So that's what the clustered means. Regularly interspaced means that the viral DNA is spaced between these repeaters. And these palindromic repeats mean that these things, these repeats, are these palindromic sequences that say to the bacterial cell, hey, this is like stuff we don't want, but these are bad guys that we should watch out for. And then there are these Cas genes that are also in the bacterial chromosome that are used in order to protect the cell when it does see this DNA again. So the way that it works is that the DNA here, the entire thing, is going to be transcribed, and so that DNA is going to make some RNA. And so here's this repeat, the palindromic repeat. Then you have originally the viral genetic material, viral DNA, and then here's another repeat and the viral DNA again, and then that gets cut, 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 cut. And so that's the CRISPR RNA processing. And so what you have is these little RNAs, and the RNAs will go together with the proteins from the Cas genes. And so anytime this is injected into the cell again, the cell can say, hey, I recognize you. I'm going to kill this viral DNA. Or maybe this is a virus that's injected itself in previously, they themselves in, then this piece of mRNA together with the protein, this is the protein, will recognize that bad new DNA coming in and will kill it. So it contains snippets of DNA from viruses, that's again this stuff, that have attacked a bacterium in the past, and they've placed it between these palindromic repeats, that's again this stuff. They're used by the bacterium to detect and destroy DNA from similar viruses attacking them, in the future. So Cas9 is an enzyme. If you go back to this part, you'll see the Cas genes. There are various Cas genes. We're going to talk about CRISPR-Cas9 right now. So this is an enzyme that cuts the DNA at the target location specified by CRISPR. So here is that RNA that we were talking about, right? Here's the palindromic repeats, and here's the viral, originally it had been viral DNA. Here's the DNA that maybe the virus is injecting, like a new virus is injecting. And do you see how it matches up here? This Cas9 enzyme now, that's what this is, Cas9. That's an enzyme that can now go ahead and cut this DNA right here. So CRISPR-Cas9 scientists use in gene editing. So here's what you can do. Let's say there's a gene right here. Let's say it's not a virus. Let's say we're using this in biotechnology in a lab. And let's say this is a piece of DNA that codes, it would be longer than this, but let's say here's a little piece of DNA that codes, maybe right here is the gene for insulin. Gene for, wow, I said that backwards. So the gene for insulin or whatever I'm interested in. Before I have to find a restriction and I hope can cut somewhere along here. Now what you can do instead of getting viral DNA, you can actually make a little piece of DNA that has exactly the complementary code of piece of DNA you want to cut. So let's say you know what the DNA code for insulin is, and that code is, whoops, that's a bad piece of DNA. Here's the insulin gene, and you know exactly the code because we can do that with other DNA technology. Let's say you know the exact code upstream and the exact code downstream. You can give, you can engineer a piece of DNA that has the exact complement to the code exactly where you want it. And so now instead of just cutting the genome anywhere the restriction enzyme is going to cut, you can tell it exactly where to cut. And then you can insert your DNA, sort of like we talked about last time with the restriction enzymes. This just gives you a much better targeting for exactly what you want to cut. And that's all I have on CRISPR, but just all the stuff that we learned before with restriction enzymes is true for this as well.